Hello and good evening. We'll be starting just momentarily. Hello and good evening, everyone. Thank you for joining us for the Canadian Liver Foundation and the Canadian Association for the Study of the Liver, a live webinar on COVID-19 vaccine safety for patients with liver disease. My name is Nem Maximovich. I am the Manager of National Health Promotion and Education at the Canadian Liver Foundation, and I will be the moderator and host for this webinar. We are joined tonight by our presenters, Dr. Craig Jenny from the University of Calgary and Dr. Jordan Feld from the University Health Network in Toronto. We will have each speaker speak before moving to a live question and answer portion. Please feel free to use the Facebook Live comment feed to ask your questions during the presentations. And please do note that the questions will be answered following both speakers' talks. This webinar will also be available post-event on our Facebook page, the page you are currently watching this video on, our liver.ca website, and our YouTube page in the coming days. So to begin, for those of you who may not be familiar with the Canadian Liver Foundation, I'd like to provide you with a brief overview of who we are and what we do. The CLF was established in 1969 and was the first organization in the world created to help people with liver disease. We are committed to promoting liver health and improving the prevention, diagnosis, and treatment of liver disease through our four core pillars: research, education, patient support, and advocacy. At this time, I would like to welcome our first speaker, Dr. Craig Jenny. Dr. Jenny is an associate professor in the Department of Microbiology, Immunology, and Infectious Diseases at the University of Calgary. He is an immunology and infectious disease specialist and frequent medical expert on CBC and other news networks. Dr. Jenny is also the scientific director of the Schneider Translation Lab in Critical Care Medicine, also the University of Calgary, a position that allows him to work directly with clinicians and researchers on human clinical studies. I will pass it on to Dr. Jenny.
Thank you, Nem. Very excited to have the opportunity to participate tonight and to show you a little bit of what we've learned about COVID vaccine over the last several months. And a little bit of the background as to how we got to where we are today, how these vaccines work, and maybe even what the future holds with regards to vaccines. So I specifically am not going to address liver disease, and I know that we will be covering that a little bit later, but I thought I'd stick with a higher level view of simply how the vaccines work for all Canadians and what we went through to, in less than a year, have a working vaccine against the first human coronavirus. So with that, just thought I'd put up disclosures related to either industry work or work with COVID, a number of CIHR team grants. The only one that might have any direct impact, not on what I'm showing today, but I will just point out that we are doing a clinical trial on a nasal spray for prevention of SARS-CoV-2, so not a vaccine, but I thought I would just flag that we do work with a company on COVID-19. So with that, I put together some learning objectives for today, and these are really the topics I'm going to try and address, and I'm hoping at the end of this seminar we'll be able to understand exactly why it is we need a vaccine for COVID-19, how the new vaccines were made, tested, and how we got to this point in really remarkable time, and specifically how the new vaccine platforms work and how they educate and engage our immune system. So starting off, this is what we're going to be talking about tonight. So this is the SARS-CoV-2 virus, or the virus that causes COVID-19. The landscape of the virus is covered in these little red blobs. These are the spike protein, and we're going to come back and talk about these throughout the vaccines, because this is really the target of what our approved vaccines are here in Canada. So our vaccines are training the immune system to recognize these little red residues. So what is COVID-19? Well, it's a disease caused by a new coronavirus, and coronaviruses do infect people. We've had them for really as long as recorded history. They are responsible for things such as the common cold. but they are also responsible for other more severe recent disease outbreaks, specifically SARS in the early 2000s, the Middle Eastern Respiratory Syndrome that is currently still active in some parts of the world, and most recently COVID-19. The little cartoon I showed you, that's the entire virus, and it uses those little red spike proteins to actually stick to our cells and enter our cells to infect us. So the spike is really the utility that the virus has to stick and enter our cells. Once it's inside our cells, it unpacks its genomic material and then uses our cellular proteins basically to make more virus, enabling us to spread it to other people. And this is what triggers the host immune response. Specifically, we get a robust inflammatory response, which under the right conditions helps clear the virus. But when that gets dysregulated or becomes too strong, we tend to see the symptoms of disease. So much of the clinical disease we observe is actually the impact of our own immune system and not necessarily the virus itself. So there has been a lot of discussion over the past, many months about whether we should just let the coronavirus run basically unchecked through the Canadian population. If we could protect those that are most at risk of severe disease and let everybody else who's in a lower risk group actually get infected, we might be able to generate some form of natural herd immunity. And that is because people who recover from an infection normally have some protection against being reinfected. So this has been put forward as a potential strategy. And although on paper, this seems to have some merit, there are a number of perhaps caveats to this overall strategy. First is when we are naturally infected, we can't control the dose. And there is some evidence that people that are infected by a higher number of virus particles, so in facing a higher infectious dose, actually get more severe disease. And when we allow natural infection, we cannot regulate this. So we lose that ability to try and ensure people get a mild infection. When there is a lot of virus in the community among, for example, people at low risk of severe disease, it becomes infinitely more difficult to actually protect the people at high risk. And that's because these people that are infected in the community may not get very sick, tend to be the vectors that transmit the virus to the people we're trying to protect. As virus increases in the community, and we've seen this across the country, it becomes impossible to keep the virus out of hospitals, out of long-term care facilities, out of schools. So the idea of trying to wall off those at risk simply is not a feasible solution. No matter how careful we are, the virus seems to find a way to get into those facilities. Perhaps more concerning is we still really don't know who is quote unquote high risk. Although we know that people of an advanced age or with underlying conditions are at greater risk than the general public, there are individuals in our community that have no underlying condition that are relatively young, even under 20 in Canada, that have unfortunately succumbed to this virus. And it's impossible to predict if any one individual is at a high or low risk. Perhaps the most unsettling piece is we actually don't even know how long this natural protection will last. And I will show you a little evidence that really argues that this may be short-lived in some people. We are now seeing emergence of new viral strains and new viral variants, and these are beginning to reinfect people that may have been previously infected with one version of the virus. And most critically, even if people do not end up hospitalized or do not end up in the intensive care unit, people still get sick. And this is something we really still want to avoid. We want to mitigate the disease burden in the community as best we can. Again, people tend to throw the simple argument that this is just another flu, that we don't have much influenza this year, and really COVID-19 is this year's flu. And I'd like to sort of counter that by looking at the actual numbers. So this is an argument I hear weekly in my interactions with the public. And if we look at coronavirus in Canada, we're now at nearly 1 million cases. This has gone up more than 3.5 times since mid-November. So we are really into this larger phase of the virus right now. Although numbers are coming down recently, there's been a massive expansion since November. We've now lost more than 20,000 Canadians to this virus in 1 year. So again, very stark numbers. If we compare that to even a bad year of influenza, we tend to think of the flu as infecting 10 to 20%, sometimes even as high as 30% of the Canadian population. So that represents, 3.5 to 7.5 million Canadians being infected. This leads roughly to 3,500 deaths in Canada, or the loss of about 9 Canadians for every 100,000 of our population. If we look at where we are with COVID at the moment, we've only infected about 2% of our population. A fatality rate that is significantly higher, and right now we've lost more than nearly 55 Canadians for every 100,000 infected. So a almost 6 times fatality rate over even a bad year of influenza. So this disease is far more severe than flu. And if we were to allow this 2% to run through the community such that we got 10 to 20% of the population infected, the loss of life would be just staggering. Now, getting back to the other problem, and that is, how long does our actual immune memory last? Well, we're starting to actually get a much clearer picture of this. What we are learning is that although people develop immunity to SARS-CoV-2 or the virus that causes COVID-19, not all of our immunity is the same. And that is, people will develop antibodies. So those markers we look for in the blood test, but not all of those antibodies are protective. So they can actually recognize different targets on the virus, different pieces, for example, of those little red spike proteins, but they don't actually neutralize the virus. So even though our immune system sees it, the virus can still infect us. We are also seeing more than a few cases now where even as early as 6 weeks after somebody recovers from symptoms, we cannot detect antibodies in their blood anymore. So this suggests that even if you build an immune response, it may not last. And in fact, if we look at clinical case studies, we are seeing more frequently now clear cases where somebody was infected, cleared the virus, and then was reinfected later by another round of virus. So there is very clear evidence in the community that just because you were infected does not mean you are now protected. So this combination of a partial or substantial failure of natural immunity and this higher rate of morbidity associated with COVID-19 really argues for the need for vaccination to help control the pandemic. But there are a few problems. There's currently no approved coronavirus, or prior to this pandemic, there was no approved coronavirus vaccine for humans. And although we had been working on it for years in veterinary medicine, we had really not seen a lot of success. So given those problems, how did we get to where we are today? Well, we were fortunate that we weren't really starting from scratch. We had a good basis for which to build these vaccines off of. And we can look at a number of different parameters. So for example, with virus information, although the virus was only identified in December of 2019, the genetic sequence, the entire genetic fingerprint of this virus, was made public by January 10th of 2020. So almost instantly, we knew exactly what this virus was, what it looked like, and what its genetic sequence was. Critically, it was important to other viruses we had been developing vaccines for. So very similar to SARS, very similar to MERS, and we could use our experience from those. With regards to building a brand new vaccine platform, many of the pieces were already in place. They had just never been brought together. So new technologies such as mRNA vaccines were being developed, but they had to be brought together with the people who were studying, for example, coronavirus. We had experience in engineered viruses, which is an interesting and exciting vaccine platform we'll talk about at the end of today's talk. And in fact, it was a Canadian-led effort that developed the Ebola vaccine, the VSV-EBOV, which is an engineered virus. And many of the pieces of our vaccines had already passed phase 0 and phase 1 clinical trials, so we could pool resources from things we already knew were safe. Perhaps critically was money. It often takes billions of dollars to take a vaccine from concept to full clinical trial, and governments stepped up. Instead of trying to pick a winner, trying to pick the most likely candidate, they funded a number of platforms in hopes of achieving at least one success And as we've seen now, perhaps many successes. And one thing that cannot be overlooked is just the support of people. It often takes us years to get enough volunteers for a large phase 3 study. And in fact, people recognized the urgency of this and signed up in large numbers so that we were able to complete phase 3 trials with 30 and 40,000 volunteers in a matter of months. So we had the public support, we had the public mobilization to make these studies happen. Now, often this process is drawn out, sequential, and takes many, many years, where you have to prove an idea, fundraise, test it a little bit, fundraise, do a larger study, fundraise. And what we were able to do for this particular approach is basically do all of these things in parallel. So there was no need to demonstrate to investors that you had a novel technology. We funded good science and strong proposals. And at the end of the day, we have seen a number of camps with success. Now, people often wonder, if we did it so fast, obviously we sacrifice safety. And I think we need to really stress that by doing things in parallel, sped up the front end of these development. We did not allow any company to cut corners on safety. So full phase 3 clinical trials were completed with 30 to 45,000 patients per trial. This is on par or in excess of what we normally request. We had complete transparency in adverse effect reporting. And in fact, we saw a number of cases very public in the media where adverse effects were reported and trials were stopped and investigated to determine if those adverse reactions were due to the vaccine or not. This, again, transparency is really unheard of, unparalleled, and should provide a confidence in that these studies were quite thorough. And finally, perhaps the single aspect that I take the most comfort in is that as a country, we are not simply following the approvals of other nations. We instead demand our own health and safety standards be met, and all vaccines must be approved in Canada by Health Canada by our standards. And we currently see that there are vaccines approved by other countries, even countries we would normally classify as allies, that we have not yet approved here in Canada because they have not submitted the appropriate paperwork and safety and efficacy documentation for us to make an informed decision. So because of this, I am very confident that when a vaccine is approved by Health Canada, it is safe and it does work. And I will then happily receive vaccine when it's my turn. Anybody who would like to keep tabs on how these vaccines are progressing through various clinical trials, the New York Times has this unbelievable resource. It's updated daily. It tells you all of the vaccines in development, including ones that have not worked and have been withdrawn or abandoned, the number of approved vaccines, where they are approved, and critically, when you click on each vaccine, it tells you specifically how they work, what their formulation is, what the technology is, what the platform is. It's a fantastic resource, and as I said, it's updated daily, so lots of information here for people that have questions. So moving on to exactly what these vaccines are, we've got a lot of potential candidates. Fortunately, we've got 2 already approved in Canada. So I just thought I'd lay out in simplest terms how these vaccines work, what their advantages are, and perhaps some of the disadvantages with these various platforms. So perhaps the simplest is to build this vaccine as we build our current vaccines, either using a dead virus or simply with 1 or 2 of the proteins out of the virus injected with an adjuvant, such as we do with influenza. So with this approach, these are the simplest, and I use simple in sort of quotes. Nothing is ever simple, but this is building off technology we already know how to do. This is using our classic vaccine strategies. In general, they're pretty easy to make, they're cheap, we can mass produce them. We tend to get really good antibody responses or humoral immunity. We tend to get fairly limited cell-mediated immunity, T-cell responses. And this is important because we've started to learn that really the quality of our T-cell response is critical to fighting off COVID-19. Often they tend to lack precision. So although they generate good antibodies, there's a whole mix of antibodies, both neutralizing that can prevent infection, as well as other antibodies that will still recognize the virus, but are not very good at stopping the infection. We also know that with these simple approaches, the duration of immunity can be pretty short. If we think of many of our simple vaccines, we do require boosters, and we require, for example, tetanus. We need a shot every 10 years because the memory for that vaccine fades. And I guess the largest problem with this approach is the results have been quite variable. So depending on which specific formulation we're talking about, efficacy data ranges anywhere from 50 to 90%. So really good vaccines to vaccines that are really not that protective, suggesting that there is a lot of person-to-person variability, a lot of study-to-study variability, and really it does put a little bit of a damper on the enthusiasm for this kind of platform. The next on the list are the mRNA vaccines, and these are the two vaccines right now that are approved in Canada, and these represent a significant step forward in vaccine technology. So this is a new approach to vaccination in humans. This is the approach used both by Pfizer and by Moderna, and recently there is a new clinical trial started in Canada by Providence, and that's using, again, an mRNA-based platform. What this strategy involves is there is no actual virus. There is no pathogen involved. We simply take a genetic sequence out of the virus and we introduce it to our body's cells. So we are giving our cells the template to make a viral protein. The advantage of this is we can actually tweak these sequences. We can optimize them. We can make them enhanced expression or modify key amino acids to make them more immunogenic. The two that have been approved in Canada both use the whole viral spike protein. So that red blobby thing on the surface of the virus, that is the gene we've included in these mRNA vaccines. And perhaps the biggest advantage is because this is made by our own cells, we generate both neutralizing antibodies, but critically we get that cell-mediated immunity as well. So how do they actually work? Well, we take an mRNA sequence. So this is a copy of the genetic material, a very short-lived temporary copy. And we stick it inside a little particle made up of fat droplets, lipids. And what these lipids do is they allow this RNA to get into our cells. So it allows it to pass into our cells and help stimulate overall immune activation. All of the components, though, are very unstable. This is why we need to store them at -80. So they have a very short half-life. Once they're injected, our body breaks down all of the components, both the nanoparticle as well as the RNA, quite rapidly. So there is no risk of long-term effects in our body. Now, looking at this from a schematic point of view, here's our body cell. We have inside that a nucleus. This contains our genetic material. And you're going to see that this vaccine technology does not influence or does not contact this at all. And what we're going to use for the vaccine is an mRNA, so a little piece of genetic material, and we're going to wrap that in this lipid nanoparticle. And when we inject this into the patient, this lipid coat actually allows the RNA to pass inside our cell. And here it stays outside of the nucleus, so there's no risk to our genetic material. And what we do is we use our own body's proteins to read this template. And basically we read it over and over and over again, and we keep making our own version of a spike protein. So once again, no virus present, no risk of infection, but now we have the viral protein. Some of this is released from the cell, and this can be used to generate those antibodies. So the humoral immunity that we test for in your blood test. So we get our neutralizing antibodies. But critically, because our own cells are making this, we can actually show this to our killer T cells, and we can build this cytotoxic cell response. So these killer T cells are also generated, and we get both parts of the immune system. Once we do this, this mRNA is very unstable, and we in fact have enzymes in our body that are designed to break this down. So we digest away and destroy that template. Likewise, when we are no longer making new spike proteins, those eventually break down too in a matter of days. And what we're left with are our memory immune cells ready for the real infection with no residual effect of the actual vaccine. So this approach has a number of advantages. As I said, we can engineer that genetic material to make them better and more precise. Critically, as we're seeing new strains and new variants approach, we can change that mRNA very quickly, really in a matter of only a few weeks if necessary, and create a whole new vaccine against a new viral variant. As I showed you, it generates both antibody and T-cell immunity, critical defenses for a coronavirus. And we can deliver either the entire protein, as we're doing with the spike protein, or we can actually pick our favorite parts of those proteins and engineer it to only generate an immune response against the specific targets we want. Now, although there are huge advantages, there's obviously disadvantages too, and we've seen this already. They're very difficult to make. This is not something that every vaccine facility around the world can make without significant upgrades. So we are limited by which companies are able to produce this formulation. We've seen the issues with ultra cold storage. So even the Moderna vaccine does require -80 storage. It's just, it can be distributed at higher temperatures for longer periods of time, allowing us to get it further out in the community. But these are not stable vaccines that can simply be on the shelf in your local physician's office. And unfortunately, both of these do still require 2 doses. So that creates the need for more vaccine, but also the logistics to give every Canadian 2 shots. Now, the final type I wanted to talk about is engineered viral vaccines. And these I find are very exciting. And we do think there may be a couple of these approved in the coming weeks here in Canada. So there are 2 general approaches here. And these are the vaccines that are currently being developed by AstraZeneca, Johnson & Johnson, but also we have some Canadian grown examples at the University of Calgary and University of Ottawa. And we are taking 2 strategies here. One is to take a harmless or relatively harmless virus, something that does not cause disease in humans, to deliver a SARS-CoV-2 gene. So very, very similar to the mRNA vaccines, but instead of a lipid nanoparticle, we're going to use a virus as a Trojan horse to get that gene into our cells. The other strategy is we can actually take the same harmless virus and we can dress it up on the outside to look like the coronavirus. So this is a technique called pseudotyping. So once again, no risk to us, but as far as our immune system is concerned, it looks like the pathogenic virus. So we're going to design an immune response against it. And I'll show you how both of these work. We are currently using viruses that are already approved for other therapies in humans, including gene therapy or cancer therapy. So we have a very good idea of the safety profile of these viruses. This same strategy, as I pointed out earlier, was used for the Canadian-made Ebola vaccine known as VSV-EBOV. And there are currently clinical trials using adenovirus as well as some other viruses in development, phase 1 and phase 2. So how do they actually work? Well, the first strategy is, as I said, very similar to the mRNA vaccine. We take a gene out of the deadly virus, so the coronavirus, and we stick that gene into our harmless green virus here, an adenovirus. And now we have an engineered viral vaccine. And this will work just the same way as the mRNA. So once again, our body cells this vaccine harmless virus, and what it does is it provides the mechanism to get this mRNA back into our cells. Just like the mRNA vaccines, once it's in, we make, for example, spike proteins and can build our killer T cells and our neutralizing antibody. So very similar approach, but instead of lipid nanoparticles, we use a harmless virus to deliver the genetic material. The alternative strategy is actually to take some of the surface proteins of the deadly virus and dress our harmless virus up so it looks like the nasty bug. There's no real risk of disease here, no risk of pathogenic infection. But when our body sees this virus, we generate the antibodies we need against the surface residues. So we are able to build those neutralizing antibodies. So for this vaccine strategy, there are again advantages. Replicating viruses, the immune system really does not like them. So it provides a very strong signal. We get long-lasting memory, just like the mRNA vaccines, they're pretty easy to reformulate. We can do that again if we need to for a new variant. They are much easier to store and distribute. They're fairly cheap to make once we've demonstrated the prototype. But again, some disadvantages. Not all patients can receive a live virus. If we think of people that are immunocompromised or other underlying immune conditions, we do not want to be infecting them with a live virus, even if it is quote unquote harmless. And we're not sure yet how many times you can receive a vaccine of this platform, because you will still generate some immunity against that harmless virus, which means when we introduce it a second time, you may neutralize it before the immune system is actually made aware of the contents of the vaccine. So it's an interesting strategy at this moment, but we don't know how many times we can use it. Looking at the big picture, where do we stand today, the beginning of 2021? We have 2 approved vaccines in Canada. And if you would have asked me even as late as September, October of 2020, would we have nearly 1 million doses of vaccine administered in Canada by the beginning of February, I probably would have said no. So I do think that this is a major achievement, a major victory that we really need to take a step back and appreciate how far we've come in 1 year. We also have a couple additional platforms that are currently under review at Health Canada, and we're anticipating approval of at least one of these in the coming weeks. And this will again enhance our vaccine supply and get vaccine out to more Canadians. Now, in the final 2 slides of my talk, I just want to, again, look back at the big picture. And although we've had stunning success with developing a vaccine that appears to be very effective and very safe, there are still some questions. And specifically, we do not know how many Canadians actually need to be vaccinated before we have a community level protection or that desired herd immunity. With things such as influenza, that can be as low as 40 to 50% of people vaccinated, but that's considering that we often have 20 or 30% of people naturally infected. For this particular virus, we think that has to be closer to 60 to 70% of Canadians. So it gives you an idea of how long it may be before we reach those levels. We've seen a lot of questions about 1 or 2 doses, timing between doses, mixing between vaccine platforms. And although we have a working strategy, we are still learning as we go along of how flexible, for example, timing can be between doses. A topic for another day would be variants. And once again, we're seeing them. We are seeing that they do influence the efficacy of vaccines. The good news is most of the variants that we are seeing are still protected to a large extent by the current vaccines. So that is reassuring. If, however, a new variant appears, we may have to reformulate the vaccines. And then finally, how long is this immunity going to last? So far, so good. People that were in the early clinical trials still appear to have immunity. But again, only time will tell. And then finally, as a summary, we still have to look at the achievement. We were able to develop the first ever approved human coronavirus vaccine in less than 10 months. And I think that is really a miracle of modern science, modern medicine, and speaks to the drive and commitment of the people that were leading these studies. Not only did we get 1 built, we actually have 2 approved vaccines in Canada, 1 additional one that is under the final stages of review. So here we are 1 month into 2021, and we may have 3 vaccines by mid-February. So again, remarkable. A good vaccine tends to give us 80, 90% protection. These first generation coronavirus vaccines are even exceeding that. So once again, a stunning success. But with all good, there are always issues. And right now we're having a difficult time getting enough vaccine into Canada. So once again, more than 900,000 doses administered. But if everybody needs 2 and we need 60%, 70% of the population vaccinated, we still have a long way to go, and we need to get this problem solved as well. So with that, I will turn this back over to Nem and to our next speaker, and I'll be happy to take questions at the end of today's session.
Thank you, Dr. Jenny, for that great talk, extremely helpful and very easily digestible information. I would now like to welcome our second presenter, Dr. Jordan Feld. Dr. Feld is a Canadian liver specialist. He's a scientist and research director at the Toronto Centre for Liver Disease at the University Health Network in Toronto. His expertise is in clinical liver disease management, antiviral research, and clinical trials studying immune response. Dr. Feld is currently appointed as an associate professor of medicine at the University of Toronto and is the current president of the Canadian Association for the Study of the Liver and sits on the Canadian Liver Foundation National Board of Directors. Jordan.
Are you able to hear me? You're able to hear me now? Sorry, by this point in the pandemic, I should be very good with unmuting myself, and I just wasn't, when I was sharing my screen, I wasn't sure how to do it, but I think I've got it now. So thanks, Nem, for the introduction, and Craig for a great introduction, well, not introduction, great presentation on everything you need to know and more about vaccines. And I would really echo what Craig said about the remarkable scientific triumph that we've seen with this vaccine development. So what I'm going to talk about is a bit more specific to the liver. I am a liver doctor, so I'm going to talk a bit more about how this relates to people living with liver disease. And so here are my disclosures. I do a lot of research, and so I do this with a number of pharmaceutical companies, primarily in viral hepatitis, Wilson disease, and a couple of other types of liver disease. And then I also do some consulting for companies that are developing new therapies for liver disease. In addition, I should say that my real disclosures here are that, first of all, I have received my first dose of vaccine. I've now received my second dose of vaccine. I do believe in vaccination, so you won't find me to be an anti-vaxxer here. And I've had the vaccine and I'm still alive with no extra body parts. So I'm saying that half in jest, but a lot of people did have concerns about the safety of the vaccines. And I'm going to talk a fair bit about that because I think it's important and I understand why people have their questions, and I think it's important to address those. So what I'm going to go through is just a little bit in terms of why we should be thinking about vaccination, is thinking about COVID-19 in people with underlying liver disease. And so I think that's really the first and most important thing about why we should be consider vaccination. And then Craig talked about how we make these vaccines and how they work and a little bit about the efficacy. And I'm really going to then focus on the safety. So how safe are these vaccines for people generally, and then specifically for people with liver disease? So, you know, it's sometimes a bad idea to mix certain things together. And this particular example, taking someone with an unwell liver and mixing it with this particular virus is a bad idea. So we know that people with underlying liver disease and people who end up going on to get COVID-19 may have a more severe course of the disease. So how do we know this? Well, a lot of studies have been done, and you've probably heard about a number of medical conditions that predispose to people to more severe COVID-19. And I think it's not as appreciated that liver disease is actually an important one of the diseases that does predispose to more severe disease. So there's now a number of studies that have shown that people with underlying chronic liver problems are associated with worse outcomes. So they're more likely to have long hospitalization if they get sick, and they actually have an increased risk of death. But I want to highlight who among that. So that's just the general category of anyone with liver disease, but I'm going to show you that it's a little more focused than that in terms of who has a higher risk than others. So, and I'll just give you an example of some of the studies. So looking at over 100,000 people in the UK, they found that people who had underlying liver disease had a 1.75-fold increased hazard of dying in hospital. So of people who were admitted to hospital. And then a smaller study, but coming out of the US, people living with chronic liver disease had a relative risk of death compared to the general population of about 3 times the risk. So that's quite a high relative risk. But what I want to point out, and I really want to stress this is mostly due to people living with cirrhosis. So for those of you who don't know what that means, cirrhosis means a scarred, damaged liver. And really what we're seeing is that it's cirrhosis that really predisposes to bad outcomes. And if you don't have cirrhosis, you're probably okay. But for people with cirrhosis, you can see that the risk of having a bad outcome is over 4.5 times greater of dying if you get COVID-19. So I'll mention a few specific liver diseases. Early on in the pandemic, it was recognized that people with fatty liver disease, very common, in fact, the most common liver disease now in Canada, may be at higher risk of more severe COVID-19, and they seem to possibly shed the virus for longer. The truth is, it's very hard to separate fatty liver disease from diabetes, high blood pressure, and obesity, which are 3 conditions that have all been associated with more severe COVID-19, and really teasing out apart whether it's actually the liver disease or it's the underlying metabolic problems that go along with obesity, diabetes, and high blood pressure is really hard to separate. But those people, certainly with both of those conditions, would be at higher risk of more severe disease. People living with viral hepatitis infection, so hepatitis B or hepatitis C, at least from what we know so far, unless they have cirrhosis, so unless they've got more advanced scarring in the liver, there's no evidence of having more severe disease because of the hepatitis B or C. And actually, there was some interest that maybe the medicines we use to treat hepatitis B and C might actually work for this virus. Unfortunately, that does not seem to be likely to be the case. What about autoimmune hepatitis? So this is a liver disease that's caused by the immune system attacking the liver. And people worry that if they have an autoimmune condition, that they might be at higher risk of having more severe disease, especially if they're taking medicines that suppress the immune system. The good thing about this is, from what we know so far, there does not seem to be any evidence of more severe disease in people with autoimmune hepatitis, including people who are taking immune suppressing medications like prednisone or azathioprine that also goes by the name of Imuran. And importantly, no recommendation to reduce immunosuppression to make the disease any milder. And Craig mentioned that much of the damage that's caused by COVID-19 when people have the illness, it's not the virus, it's not the SARS-CoV-2 virus, but it's your body's immune response. So actually, sometimes being on these medicines, although it may actually make you carry the virus longer, it may dampen the response to the virus. So there's no real suggestion that it causes more severe disease when you're on immune suppressing drugs. And then what about bile duct diseases like primary biliary cholangitis or PBC or primary sclerosing cholangitis, PSC? What we see is there doesn't seem to be any evidence of worse bile flow or other complications with these conditions. But interestingly, some people, particularly people with very severe COVID-19, have ended up with a very severe bile duct injury that looks a little bit like primary sclerosing cholangitis, and it develops as they recover. Not very many cases of it reported so far, but it can occur. But for people with these underlying conditions before they get COVID-19, does not seem to be worse or more severe in them, which is good. Now, how do we know this? Well, fortunately, we have a number of registries, and what these are doctors around the world putting information in about people with liver disease who get COVID-19. And so what this is we have one that's in North America and there's one in Europe, and this is storing completely anonymized data, so there's no way for it to be linked back to you. But it does require doctors putting this information in. So if you are seeing a liver specialist and you get COVID-19, it's worth mentioning it to them that there are these registries, because the more people that put this in, and especially if they put in everybody, not only those people who have bad outcomes, then we get a reasonable handle on what happens when people have liver disease and develop COVID-19. But let me just show you a little bit of the information that we've learned from some of these registries. So this is looking at people, 386 people with cirrhosis, a similar number without cirrhosis over a host of almost 30 countries. And what you see is that people with cirrhosis are at a much higher risk of a severe course of COVID-19 and death. So what you see here is, if you look at these bars, the higher the bar, the higher the risk. And what you see is that you see the blue bars are people with chronic liver disease without cirrhosis. And you can see that even if they end up getting hospitalized, you can see their chances of needing to go to the intensive care unit, needing to go on a ventilator, and certainly of dying are much, much lower than people who have varying severities of cirrhosis. And what you see in on the other side is that you see that people without cirrhosis, you can see that really the biggest risk for those people dying is their age. So as their age goes up, we have heard that in the general population, that certainly being over 65, and once you get over into your 80s, the risk of death is much higher. And you see what happens in people with cirrhosis is we see that this is really changing the risk profile. So even young people here down in their 20s and 30s who have cirrhosis have a non-trivial risk of death. And so that's why what I mentioned is really stressing this point that it's really cirrhosis. It's not liver disease without cirrhosis that is a big risk factor. And what do people die of if they have cirrhosis and get COVID-19? Well, still primarily lung disease, but liver disease is the next thing on the list. And one important thing to consider, and this is something to remind your doctors if you happen to have liver disease and should get ill, is sometimes people don't actually have lung problems. They actually just go into liver failure. So the same things that happen to anyone with advanced liver disease may occur in someone who gets a COVID-19 infection. And so it's important that we make sure that now when you go into a hospital, you'll typically be screened for this infection, but certainly make sure that if suddenly your liver disease is getting worse, even if you're not coughing or have a fever, that you're getting checked to make sure that it's not SARS-CoV-2 infection that's tipping you over. And this is just another study that's showing similar thing here, that really cirrhosis is the big driver, so that you can see that people who have cirrhosis, these are people who actually ended up in hospital to begin with, and you can see almost a third of the people with cirrhosis ended up going on to die, whereas of the other people in hospital, only 8% died, so a much lower risk of death. And what we know is that the more severe the cirrhosis, the more severe the outcome of COVID-19. So in other words, it's like any other infection. We know that infections are bad in people with cirrhosis. So when people with cirrhosis do badly with infections, and people with very bad cirrhosis, so more advanced liver disease, do very badly. And that's really probably the most important point about thinking of the consequences of this in people with liver disease. And this is really a very consistent finding. I showed you a couple studies already. This is another one where they looked at people who had COVID and cirrhosis and compared them to people who had just COVID alone or just cirrhosis alone. And what you see here is that the people who had cirrhosis alone, actually cirrhosis can be bad. So you can see they didn't do much better than people who had COVID-19 and cirrhosis, but both groups did worse than people who just had COVID-19 alone. So it's really highlighting that the liver disease, and particularly cirrhosis, is the big problem. Now, I mentioned immune suppression, but I just want to really stress this point. We get a lot of questions about this for people who are on medicines that suppress their immune system. There does not seem to be any association with worse disease in people who got the original SARS virus, the MERS virus, the Middle Eastern Respiratory Syndrome, or COVID-19. I mentioned autoimmune hepatitis. We haven't seen any concerns there. And we've also seen small series of people who have had liver transplants and other organ transplants. And although some data have suggested that they might have slightly worse outcomes, in truth, when we put most of the data together, it looks like the outcomes are actually pretty similar, even in people who have had a transplant. What we do see is that they tend to carry higher virus levels. Because the immune system is a bit dampened, the virus is able to replicate to higher levels without being controlled by the immune system. And that means they might be at a higher risk of shedding or spreading to other people. So it's very important for people that have a suppressed immune system that they're doing everything they can, all the social distancing, masking, careful public health measures, to reduce their risk of getting infected. And importantly, they shouldn't reduce or avoid required immune suppression. This can actually lead to a flare of their disease. And even if they get COVID-19, although some doctors will reduce the dose of that, there's really no evidence to support that approach. So to summarize, COVID-19 may be more severe in people with chronic liver disease, but the greatest risk is really cirrhosis from any cause. Doesn't matter how you got it. If you have cirrhosis, the risk is higher. Immune suppression does not seem to have a major effect on the risk of severe disease, so make sure not to stop any immune suppressing medicines that you've been on that are controlling disease. So what about the vaccine? Well, there's certainly been a lot of questions about this. People have asked, could this mutate your DNA or increase your risk of HIV? Maybe it was rushed. Maybe Bill Gates is planting a microchip inside your head. These are all things that you can find people complaining about, but then there are other potentially more serious scientific concerns, things like antibody-dependent enhancement, where giving a vaccine could actually make the infection worse. On the flip side, you hear scientists like Craig and I talking about this as a scientific triumph, that it's very safe. The vaccines are lifesavers. Trust us, we're doctors. These are all the types, and you get this sort of a bit of a battle sometimes in social media and in other places. And I think it's important to look at the data carefully. So I'm just going to show the data from one of the vaccines, from the Pfizer-BioNTech vaccine, which is available in Canada. And this was the first one approved here. And just to show you, they are taking safety very seriously. Craig made the point that these have gone through rigorous phase 3 trials, like any vaccines, in some cases bigger than usual. So this is just looking at what happened. They had over 21,000 people that received the vaccine or received a placebo. And they followed these, the doses were 3 weeks apart. They followed everybody very carefully in those first 3 weeks. And then they follow up everybody for 2 years. They'll do an active surveillance period, and then which is for 2 months, and then they will continue to report more serious adverse events out to 6 months after the second dose, and then continue to follow people out to 2 years after their second dose to find any more serious complications. So a pretty rigorous safety plan with lots of people. And I just want to show you what they reported in terms of the safety. And this is a bit of a complicated figure. I'll try to walk through it here. So if you look, first of all, if you look at, this is divided into the first dose, the side effects people had, and the second dose. And what you see are, first on the left are the local side effects. So just like most shots, if you get your shot in your arm, and this is an intramuscular injection, so it's going deep into the muscle in your arm, people do have side effects. You can see about 80% of people in the vaccine group had some pain in their arm. These usually lasted about 2 to 3 days at the most. And you can see that having other side effects like redness or swelling, shown here at the bottom, were quite rare. What about more serious side effects? Things like having a fever, or feeling lousy. You can see fever was actually very rare after the first dose, a little more common, 16% after the second dose, but people did feel a bit lousy, particularly after the second dose. You can see about half of people had some fatigue, had a bit of headache, felt kind of yucky, and about half of them took medicines to help with that, something like Tylenol. And what you see is that, interestingly, in older people who arguably need the vaccine more, you can see similar local reactions, so the pain in the arm and stuff, but they actually tended to have slightly milder side effects in terms of things like fever, fatigue, and headache. And what we hope that doesn't mean is that the immune response to the vaccine was less, but at least from what we know so far, the protective antibody levels seem to be pretty comparable. So some other side effects that people reported, small numbers of people, of just under 1%, reported that they had swollen glands, or the medical term for that, lymphadenopathy. And one thing that certainly raised some alarm bells was that there were 4 people who got the vaccine who had Bell's palsy. And Bell's palsy, you may have seen people with one side of their face that was paralyzed, and it recovers to some degree. Nobody in the placebo group. But it's important to remember this is 4 out of 21,000 people, so quite infrequent. And you can see this is the time that it happened after vaccination, 3, 9, 37, or 43 days after the vaccine. And normally you see about 23 cases of this per 100,000 per year. So just to compare that in terms of the background rate would be this number, 0.00023, versus what was seen in the trial, 0.00021. So really similar to the background rate. Now, is it possible that this increases the risk of Bell's palsy? It's possible, but it's very rare. And interestingly, we haven't really seen it reported since the trial was completed. So certainly something they're watching for, but doesn't seem to be a big concern. Guillain-Barré syndrome and other neurologic problems, which very rarely happen with things like the flu vaccine, have actually not been reported with the mRNA vaccine so far, which is very reassuring because these are serious but rare complications of some other vaccines, which we haven't seen with COVID vaccines. And actually, they haven't really seen any other concerning treatment-related serious side effects. There were 6 deaths in the trial, 2 in the vaccine group, 4 in the placebo group, and all seem to be entirely unrelated to the study. Now, what about allergic reactions? Many of you may have heard that after the vaccines were first evaluated in the UK, 2 cases of anaphylaxis were reported very early. These weren't in the trial, they were after approval. Both were in people with known severe allergies, and both of these people actually carried EpiPens regularly. But since then, this has been followed quite carefully. And so far, there have been 21 cases out of almost 1.9 million people vaccinated, giving a rate of about 11.1 per million people. And actually that record was a couple weeks old. As of January 18th, they've reduced that to 5 per million cases of anaphylactic reaction. Doesn't mean it never happens, and that's why they'll observe you for 15 to 30 minutes after the vaccination to make sure you don't have this. And it doesn't happen only in people with a history of allergies, but primarily. The Moderna vaccine, the other mRNA vaccine, seems to have a slightly lower risk of 2.5 per million doses. So these are very rare things and seem to be reactions to other components of the vaccine, possibly the polyethylene glycol that's the carrier of the vaccine. Now, just to highlight what that risk actually entails, we don't usually think of the toilet as a dangerous activity, going to the toilet, but severe allergic reactions happening about 2.5 to 5 per million. Just to compare that, toilet-related injuries that result in an emergency room visit are about 225 per million per year. So I'm saying that in jest. I don't mean to trivialize the risk of anaphylaxis, but I just want to highlight that the risk is really quite low. What about long-term safety? Well, vaccine reactions typically happen very early. They usually happen within days to weeks after a vaccine, and very rarely beyond 2 months. That's why the FDA is doing this very close surveillance for 2 months after the vaccine recipient in the trial, the people received the vaccine, to really follow them closely. We now have millions of people who have had more than 2 weeks of follow-up after 2 doses of vaccine, and we really haven't seen problems occurring. But there is very careful ongoing data collection all over the world in many, many countries. Is it possible that there will be delayed safety issues? Sure, anything is possible, but it's very unlikely. We really have not seen that with other vaccines, that late problems occur months or years after vaccination. What about for people with liver disease? Are there any specific concerns? Not that we know of. There were small numbers of people with chronic liver disease in both trials that have been reported so far, with no safety concerns reported. And also with the AstraZeneca vaccine, there were very few people with liver disease in that study, so we can't say too much about it. Will immune suppression or the liver disease affect the response? It's possible. We had very few cases, so we don't know whether the effectiveness of the vaccine will be lower in people who are taking immune suppressing medicines. But even if it's a little bit less effective, it's still more effective than without vaccination. So we would still generally encourage people to get vaccinated. And Craig made the point that the live virus vaccine should not be given to people with impaired immunity. So if you sort of look at this as the risk versus benefit, the vaccine, clearly there are some side effects that happen in the first few days, especially after the second dose, but they're short-lived. Serious side effects seem to be very rare. Late side effects are unknown, of course, at this point, but are unlikely given that we haven't seen them occurring yet. And it does seem to be very effective at preventing at least symptomatic infection. Now, what are we preventing? Well, for many people, it's likely to be a mild disease, even if you are, especially if you're young and healthy. But we know that the risk of severe disease is higher in people with liver disease, especially those with cirrhosis. I showed you that, and I hope I convinced you that for people living with cirrhosis, this really is a bigger concern, and so the vaccine is quite important. The other thing to remember is that even if you don't have severe COVID-19, some people end up with a slow recovery, this sort of long hauler syndrome, and that can happen even in people with a mild course of the initial infection. And the truth is, there may be long-term complications to having had this infection that we just don't know about yet. So when you put these together, the risk of COVID-19 with liver disease is certainly a greater risk than any potential risk of what we've seen with these vaccines so far. So should you be vaccinated? I would certainly strongly encourage you to be vaccinated for your own protection, because I think the risk versus benefit definitely favors vaccination, and also to protect your colleagues and family. Part of our responsibility to each other really is this. We've been saying for a year now that we're all in this together, and I think there is, just as people participated in the trials, I think getting people vaccinated will be important to bring down the disease burden and really save our healthcare system as well from being overwhelmed. So to finally, just to finish, we have put together some guidance about the vaccine. This was developed by a number of liver specialists and endorsed by the Canadian Association for the Study of Liver, so CASL, and by the Canadian Liver Foundation. It's on the Canadian Liver Foundation website under the We Are Liver section, and there's a PDF, and it goes through some frequently asked questions and some important points, most of which we covered today. And I'll finish with that, and hopefully take some time for questions. So I'll stop sharing, and hopefully we've left a little bit of time for questions.
Yeah, we have a few. I mean, we were going to end at 8, but we can probably take about 5 minutes or so if you guys are ready for just a few questions. You definitely covered a lot of the aspects, Dr. Jenny as well, Dr. Feld as well. So we're going to look at some of these questions here. And Dr. Jenny, if you haven't turned the camera on, we can turn all the cameras on for all the speakers. Okay. So we'll go to this one camera, and I'll leave it at for either of you to answer here. It's kind of, I guess some people with liver disease are generally worried that the COVID-19 mRNA vaccines may worsen their liver disease and they may opt to not getting it. I guess some of the questions are, how can they be confident that there won't be any long-term side effects?
So, I mean, maybe I'll start with that. I mean, I mentioned that you know, we've been using vaccines now for decades, and the risk of late complications from vaccines is exceedingly rare. There's not really good evidence for any vaccines causing really late delayed problems. You've undoubtedly heard things like the autism controversy. That is not a real phenomenon. It's been completely debunked. So people are not developing autism years after getting the measles vaccine. Unfortunately, it persists to be a meme on social media and people don't get vaccinated. But what I can tell you is if you've seen measles and you've seen people die of measles, you'd be getting the measles vaccine. That's really the only example that people have really erased of late toxicity from vaccines, and it's been really debunked. So I would, you know, ask Craig as an immunologist, any reason that you would expect to see any kind of late complication?
No, not at all. And in fact, we have to remember these mRNA vaccines, the target in them, the mRNA itself is something our cells naturally produce millions and millions of copies of every day. So our cells are designed to handle that exact template and use it to build all of the proteins normally in our body. So there is really no risk to altering our cells, altering our DNA, or causing any permanent damage. It is simply telling our cells to make yet another protein, and it happens to be a protein that our immune system learns to recognize.
Perfect, thank you. And I guess kind of a follow-up to that one was a question in the feed regarding some of the shortages between the first dose and the second dose. Would there be any effect, or does that lower any of the effectiveness of the vaccine if a dose isn't given in that 21 to 28 days and perhaps a few weeks after that?
I'll maybe start with that and then hand it over, but we do know the doses can be fairly flexible. So the guidelines have now added a few additional weeks in there, and those appear to be very efficient still when receiving that second shot, even 5-6 weeks after your initial shot. We believe there's still even more margin for flexibility, but it hasn't been tested. So the good news is they are somewhat flexible. We do have that extra bit of buffer time. We don't want to see that extended out to 9 months or a year between shots, but a couple additional weeks is not going to make a big difference in the overall efficiency of this vaccine.
I don't really have much to add to that.
Great. We'll just give a second last one here. I know, Dr. Feld, you mentioned some immunosuppressive activity with liver disease. Anything for some of the pre-transplant or post-transplant patients who have reached out to us with respect to their system potentially being more immunocompromised than some of the other individuals?
So pre-transplant, no. So I mean, what I would say about people are, I don't know, it looks like my video is is on the fuzz there, but can you hear me okay?
Yeah, you're good.
I just turning off the video for a second. I'll put it back on. There we go. So yeah, the one thing that I would highlight for people that are waiting for a liver transplant, this is the group that's at the highest risk for severe COVID-19. So, and we've certainly seen this in the hospital, where when people with very advanced cirrhosis, who are usually the people waiting for a transplant, get COVID-19, these are the people that get very sick. And it's often not that it's not always that they're going into liver respiratory failure or breathing problems. It can tip their liver disease over, almost like any other infection. And so this is a group that really needs to be prioritized for vaccination. And obviously, do we have information about the safety of these vaccines in people waiting for liver transplants? No, not yet, because we can't study every population all at the same time. But this is a group of people where I would strongly recommend they be vaccinated because any small risk that the vaccine might be harmful in that group is hugely outweighed by what we know is a major risk of this infection requiring.
Jenny, anything to add on your end?
Yes, I mean, again, unfortunately, these more severe groups are typically not included in our healthy patients for the large phase 3 clinical trials, and we learn as we go forward. But what we do know about other vaccines and just the general immune system, there's no reason to believe there would be any increased risk in these patients about getting vaccinated. Again, unfortunately, not really the number of patients or the time to have determined this clinically, but again, understanding the mechanism of the vaccine, how other vaccines work, there's nothing that would make me suspicious that there would be an unexpected impact of vaccinating these patients prior to transplant.
The only thing I would comment is the vaccine might be less effective in people with very advanced liver disease. We know people with cirrhosis often don't respond to other vaccines as well, so it is certainly possible, but I would make the case that any reduction in the severity of the disease, even if you have even if it's not completely protecting you, if you can reduce the severity of the disease. And the example that I would give to many of you, you've probably had the flu shot and still got the flu, and it's much milder than if you don't get the flu shot and get the flu. So this would be the same idea here, that if we could reduce the severity, we might reduce the complications of getting COVID.
And even the starting point of these vaccines at 90 to 94%, we've got a lot of room to lose some efficiency and still be quite good vaccines by standards of other vaccines we use routinely.
Great, thank you. I'll just ask one last question here. Perhaps both of you can kind of chime in on it. Broadly speaking, from both of your clinical research and public health perspective, how has or is this pandemic or has affected the care of people with liver disease, kind of generally speaking?
Yeah, maybe I'll start with that. I mean, it's certainly been a challenge, I think, for all of us working in healthcare this year. I mean, I think this year has been difficult for everybody. It's been definitely very difficult working in healthcare, and for many reasons. I mean, we've tried to adapt, but people nervous about coming to the hospital. They're afraid of getting care. But I really want to highlight to people that it's very important that you continue to get the care that you need. And I really can't stress that enough. There's a very poignant case that sticks in my head, which is very upsetting to me, of someone who was supposed to get their routine ultrasound as screening for liver cancer, and someone with cirrhosis missed an ultrasound because felt that it was too risky to get it back in the spring. And certainly I understand that concern, didn't get it done. And unfortunately, when they finally did get an ultrasound done in December, had a very advanced cancer that is no longer curable. And that obviously is the worst case scenario. I'm really speaking from that's a true story and a personal experience for me, as that's very upsetting when that kind of thing happens. But I would just really stress to people, healthcare facilities are very careful. They're not perfect, but they're very careful. And if your doctors and nurses and other healthcare providers are telling you that you need certain things done, I would strongly encourage you not to avoid healthcare facilities out of fear. I hope that I can't make that message strong enough, but we've tried to adapt. We're doing a lot of virtual care and things, but some things still require people to come to the hospital.
Yeah, I probably can't echo that more strongly. And whether it's liver or any other disease, hesitancy to seek medical attention, unfortunately, has often severe consequences. So the other side, though, I think I see quite frequently, at least in the conversations I have, is there is a significant mental health impact of this too, and worry about catching the disease, worried about catching the virus, worried family members, worried about bringing the virus home to a patient. And these are real concerns, real issues, but it's also important to realize that, worrying about them is normal. There's nothing wrong with you if you're worried. Talk to somebody. Mental health is very real. And I think you'd be shocked how many people are struggling with the overall concept of COVID-19 in the community. And that's often even with people that do not have underlying medical conditions. So if you're stressed, if you have concerns, please reach out, talk to somebody. There's lots of resources and lots of people willing to try and help you to the best of their ability.
And just on that note, I mean, we've certainly unfortunately seen with some of the mental health stressors, people struggling with things like addiction, which sometimes had been under pretty good control for a long time, and the stress of all the things that have gone on in the past year have tipped people over. And so again, I would just encourage you, if that has happened, get the help you need. We are certain, I can tell you, as healthcare workers, very aware of this, and as Craig mentioned, there really are resources all over the country to try to help people through some of these very difficult and very real challenges that we're all struggling with.
Perfect. Well, thank you guys for that. With that final note and that final answer, that concludes our webinar for this evening. I would like to take this time to thank our two speakers, Dr. Craig Jenny, Dr. Jordan Fell, for the great presentations. I also want to give a special thank you to our partners at the Canadian Association for the Study of the Liver for supporting this webinar tonight. And most importantly, all the participants, listeners, and our entire Canadian Liver Foundation community for tuning in. As mentioned at the beginning, this presentation will be available on our Facebook page, our liver.ca website, and a CLF YouTube page over the next few days. And to learn more about COVID-19 vaccine safety for people living with liver disease and all other liver diseases, or to find out about the Canadian Liver Foundation and our mission in bringing liver research to life, please visit our website at liver.ca. And please be sure to follow us on our social media, or Facebook, Twitter, and Instagram channels for all the most up-to-date information and liver health tips. Once again, I thank you all for attending, and we hope you have a safe and wonderful evening.